We've talked about evolution. We've talked about Darwin's theory of natural selection. We've talked about Mendel's work in genetics. And yesterday we started talking about taxonomy, how organisms are classified by who they're related to. And what we're going to do today, start today, is a lab where it's going to be a culminating activity. It's going to address genetics, taxonomy, and evolution.
Like towards the bottom part is like darker.
Okay. Mike?
Somewhat different sizes.
Okay, size is a variation. It's hard to tell just from photos, and it's hard to tell from just a small sample. If you went out into the rainforest and saw a couple monkeys and one was big, one was small, can you just rely on that? Now, when we did the possum lab, did you just measure one possum's ears and one possum's feet? No, you collected lots of data. There's two types. There's gray-fronted and there's red-fronted. Now, this is hard to see, so I'm going to highlight it for you here. This here is the red-fronted's distribution, and this up here is even lighter. That is the gray-fronted's distribution. We want to preserve the species. So, and we've talked a lot about zoos, and when we went to Brookfield Zoo, we made sure when we looked at different species, we knew what their niche was, we knew what their habitat was, things like that. We want to keep these species pure. Very good. Okay. So gel electrophoresis will separate the fragments out, and then it'll give each species a pattern. Now, when you look at this one, and this one's beautiful because guess who did it?
You?
Yeah. It's the nicest one. But this is species A, B, C, D, and E. This one was the question mark.
Science and everyday life cannot and should not be separated.
Rosalind
Cream
Franklin.
Beautiful.
Oh yeah.